This semester, we had the amazing opportunity to learn how to perform aseptic brain surgery on a rat.
The first part of the semester was dedicated to learning the surgical techniques. We learned how to make one deep incision using a scalpel. Then we practiced how to suture, mostly using simple interrupted stitches, on a pig's leg.
Finally, we were able to meet our participant, a white rat we call Dude. Since we have never handled a lab rat before, we learned the basics in how to handle a rat.
Then the fun stuff came next, pre-surgery training and brain surgery. Before we jump into our own experiment, let us explain more about the brain region called the hypothalamus. The hypothalamus controls the 4 F's of psychology: feeding, fighting, fleeing, and mating. We focused on the ventromedial hypothalamus, or the VMH. This helps control hunger. The VMH inhibits hunger, meaning it stops you from feeling hungry all the time. When the VMH is lesioned, lab rats can eat and eat until they die from obesity.
There had also been research on how the VMH affects food-motivated tasks. Previous studies found that rats with VMH lesions could remember tasks they were preoperatively trained in, but could not learn a new task. However, these studies used 2 different groups of rats to test this. One group was the remembering group and the other was the learning group. None had tried this theory on a single rat.
This is where our study comes in. We pre-trained our rat in a food-motivated task in the Skinner box. We trained him to push the lever for food only when the light was on.
This training took 1 week with 30-minute training sessions a day.
First, the rat had no idea what the Skinner box did. We trained him to find the pellets from the food tray. Second, we left the light on and let the rat figure out that every time he hit the lever, he got a food reward. Finally, we used a 1-minute light on, 1-minute light off intervals. At first, the rat was so frustrated he wasn't getting any food when the light was off. However, he learned quickly only to press the lever when the light was on if he wanted a food reward.
Finally, after 1 week, we ran a 25-minute trial. The rat hit the lever 240 times when the light was on, and only 8 times when the light was off.
Next came the coolest part. We successfully performed surgery to lesion the rat's VMH.
It's not gonna.
You're about to break through. After 2 hours of surgery, he was not a happy camper when he woke up. Almost there.
After 2 weeks of recovery, we put him back in the Skinner box to see if he remembered it. He hit the lever 161 times when the light was on and 6 times when the light was off. It is clear that he remembered it since he had about 3% error rate both pre and post surgery.
The final stage of the experiment, we tried to train him a new food-motivated task. We trained him to push the lever for food when the sound of a clock came on. Despite the same training schedule, the rat was unable to learn a new food-motivated task.
Our findings support that VMH lesions inhibit learning of a new food-motivated task, but don't affect remembering all food-motivated tasks.
After collecting all of our data, we had to euthanize our rat. We placed him in a carbon dioxide box and then extracted his brain.